everyone. This video is to walk you through the outside of the cell lab, and this lab is going to begin our cellular biology section. So all the labs you've done up until now have been under our biochemistry cycle of study, but the outside of the cell is going to begin our cellular biology section, where we start talking about all the intricacies of the individual cells in the body. Now, this specific lab is going to talk a lot about the cell membrane, or the outer part of the cell. This textbook chapter will be covering called Insane in the Membrane, and there's a lot of components to the cell membrane or to the outside of the cell. So this concept is going to be very important to understanding how the body functions and things that can go wrong in the functioning. So pull up your lab procedure, make sure you read all the way through the lab before beginning, and we'll go through some of it together. I'm looking at part A of the procedure, and it says, share your thoughts. Now in this section, you don't need to research anything. It's simply to put your knowledge that you have now and to start thinking through these concepts critically with the knowledge you currently have, so no research is needed for this. So think about your body. Number 1 says, what does your body need to survive and function on a daily basis? So what are your basic necessities for life? Okay, what things do you need? What things do you need to take in? What things do you need to expel? Try to think about all the different things you might need. Now, there might be a very extensive list, but put down the most important, the most necessary things. Number 2, it says review the necessities below and briefly explain what would happen if you couldn't perform that necessity. So what would happen if you couldn't eat, if you couldn't breathe, and if you couldn't dispose of waste? Okay, what would happen? What do you think? Just put a few sentences in the boxes there in that table. Now going to number 3, it says, even though humans are made up of trillions of cells, we still have the same basic requirement as the one individual cell. So even though we have trillions of cells in our body as an organism still needs the same things that one cell needs. We need a source of energy, we need oxygen, and we need a way to dispose of waste products, whether that's the whole organism or just an individual cell. Okay, now we have trillions of cells in our body. So why do you think we have trillions of cells? Why can't we just be made up of larger cells and have fewer of them? Is there a reason, or do you think it would be the same? So why do you think cells are so small? Think about it. Write down your answer for number 3. Now, as we grow, our cells grow in size, but we also get a lot more cells. So a baby, newborn, is going to have a much smaller amount of cells in their body that a full-grown adult has. Okay, so as we grow through the life stages, go through adolescence to adults, we do grow in our cell size, but more so, we get new cells in our body. So an adult has a lot more cells than an infant. Now, why do you think that is? Why couldn't that infant just keep the same amount of cells and just let them grow very big to make a full adult? Or could that work? So think through these concepts critically with the knowledge you have, trying to reason it out, okay, and put down what you think for part A. Part B is going to start our experimentation. So we have a couple different experiments we're going to do, some graphing that's going to happen. Okay, we're going to look at it with models for those kinesthetic and visual learners. We're going to look at it in terms of graphs to help out with those visual learners, people who are more math-oriented in their thinking. So we're going to do this a few different ways to try to figure out what's happening with the outside of the cell membranes and the stuff inside the cells. So part B, you might want to pause the video, go to your lab, make sure you have all of your equipment set up. For this lab, you're going to need your beaker. Okay, you're going to need some food coloring, which should have been in your lab kit, your ruler from your lab kit. You will also need your 2 hard-boiled eggs. So this is a part of your grocery shopping for this lab, okay? 2 hard-boiled eggs. If you don't know how to hard-boil eggs, it's very easy. You just put the entire egg in boiling water for about 8 minutes. And if you don't know this nice little trick, after you take the boiling egg out of the water, put it in some ice water right away, and it'll make it really easy to peel, okay? So once you have your 2 hard-boiled eggs, you want to get that together with your safety goggles. You'll also probably want some paper towels to help clean up, and then you'll need a knife to cut your egg. Okay? So get everything oriented, let your family know you're doing a lab, make sure your counter space is nice and clear, and make sure that you have enough time to complete the lab in one sitting. Once you're ready, we'll go ahead and get started. Part B says surface area and volume. So the surface area is the outer portion of an object, just the outer surfaces, okay? The volume, on the other hand, is all of the matter, so the inside as well as the outside, okay? So think about all the particles that make up matter. The outer portions, just the outer portions, is the surface area, whereas the volume is everything inside as well, okay? So it says, just like an organism, an individual cell requires a variety of substances to be transported into and out of the cell. We need energy, we need nutrients, oxygen, and we need to move waste products. So this outer cell layer is called a cell membrane or a plasma membrane. The two words are interchangeable. And it's responsible for letting the good things in and getting the bad things out. So your cell membrane is a concept we're going to talk through because it does a lot for the body, and it really helps to maintain a lot of the homeostasis or the balance of the body. Now, the cell membrane is what we call semi-permeable, semi meaning somewhat. So a semi-permeable cellular membrane lets certain things into the cell and certain things out of the cell, but not everything. It's selective in what can come in and out, okay? Now that is incredibly important when we think of it in chemical terms because it means that the inner part of a cell can have a different chemical environment than the outer part of the cell. Pretty neat. So go ahead and collect all of your material and then we'll start doing the lab together. Okay, I've assembled all of my equipment that I need. I have everything ready to go, and so we're going to jump into part B and actually do our experimentation. So as a reminder, make sure you pull back any long hair, get rid of any dangling earrings or necklaces, and maybe you put on an old t-shirt just so you don't stain anything with your food coloring, but that's up to you. Now for part B, it says we want to place 100 milliliters of hot tap water into a beaker. So grab your beaker from your kit, find your 100 milliliter mark, it's not very much water, and you want to fill it up with hot water. Now I'm just putting the setting on my sink all the way to hot, making sure it feels pretty hot and filling it up that way. You don't need to have boiling water. And you only want 100 milliliters. Okay, so there we have it. Now we want to add 20 drops of food coloring. So hopefully everyone has a dark color. This is the food coloring that I'm going to use. I actually made my own, so we're going to see how it works. We didn't have any in my lab kit, and so I thought I'd be creative and see if this experiment works. So you want 20 drops. But hopefully anyone who didn't have their coloring told me, so you guys have everything you need. You can give it a little swirl, get a spoon, stir it, okay? So you want to color your food coloring. Now because mine is not as concentrated as some of the store-bought ones, I might add some more color into there. And that's okay to do. So if you notice that yours is pretty light in color, especially if you were given like a yellow or a pink, you can add a little bit more. So we'll see how that goes. Okay. You want to be careful, again, not to stain anything. And then you want to get your 2 eggs and go ahead and peel them. Once you peel your eggs, you're going to take a knife and you're going to cut one of the eggs into a 1 centimeter cube, which means you need 1 centimeter on every single side of the cube. So you can use your ruler to measure it out, get as close as you can, but it doesn't have to be perfect. So go ahead and pause the video and cut your egg. Okay, so I have cut my cube out of one of my eggs. And I now have another whole egg, and you're going to put both of these into your food colored water. Now I went ahead and I added some more food coloring just so I can actually get some color. But again, if all of you have your regular bottles that were included in your lab kit, 20 drops should do it. If you add more, that's perfectly fine too. Just don't put any less, okay? So you're going to take your cube and you're going to take your whole egg and you're simply going to drop them into your coloring solution, and you want to set a timer for 30 minutes. Now, if you go over the 30-minute mark, that's okay. Just don't go under the 30-minute mark. All right, so we're going to leave that and let it do its thing. And in the meantime, we're going to move on to the second part of our lab, where we'll actually be using Play-Doh. For the next part of our lab, you're going to want to get your Play-Doh out of your lab kit, as well as a container that you can pour some water into and that you can put your Play-Doh in. Now, you're also going to need to cut your Play-Doh, so I suggest maybe getting a plastic knife if you have one available, so that you can just throw it away instead of getting Play-Doh on your nice kitchen knives. Okay, so we're going to move on to our Play-Doh lab while our egg cube and our whole egg are soaking in our food coloring solution. So make sure that your timer is set and you're keeping an eye on it. Again, if you go over 30 minutes, that's okay. But in the meantime, let's go ahead and move on to our Play-Doh experiment. So for this, your lab procedure says to make 8 1-centimeter Play-Doh cubes. You're going to make a 9th cube that's a 2-centimeter cube. So 8 1-centimeter cubes and 1 2-centimeter cubes. So get out your Play-Doh, and again, make sure you have a nice knife and your ruler, and you're going to cut it as accurately as you can. They don't have to be perfect, but you want 8 1-centimeter and 1 2-centimeter. Okay, so I now have my 2-centimeter cube and 8 1-centimeter cubes. Now you'll notice your 1-centimeter cube is actually a lot smaller than your 2-centimeter cube. So just something to note. But once you have those all cut up, you can push your Play-Doh off to the side, and we're just going to focus on the cubes now. Now it says stack the 8 1-centimeter cubes into a 2-centimeter cube. So we're going to stack all of our 8 cubes together, and it will look similar to the size of our 2-centimeter cubes. Okay, don't smush them together, just simply set them all on top of each other. And you're going to have to put 4 cubes on the bottom and then 4 cubes on the top. Again, don't smush, just set. And if your Play-Doh cutting skills were anything like mine, they're a little bit malformed. But we roughly now have a 2-centimeter cube made out of our 8 1-centimeter cubes. Okay? Again, don't smush, just set them there. Now, once you've done that, you want to place the cubes, both your 2-centimeter and your 8 1-centimeter cubes, into a container for water. Okay, just some kind of small container. It doesn't have to be clear. And you can actually put them both in the same container. Just gently set them in. Okay. Now, once you set them in there, it says that you want to add water. You can do this in a couple different ways, but you just gently want to pour water on top. If you have them both in the same cup, you're just going to pour water. Make sure you touch both of them with your water. And you're going to pour it so that each of your cubes are submerged. So I'm just gently pouring it over both of my cubes, filling up my container. Okay. Now my cubes are both submerged. I was pouring back and forth on both of them gently. And we just need to leave them in there for a few seconds, and then we're going to observe what happened. So you've poured your water. Now you want to slowly take your cubes out and observe. So in the box below where it says observations, look at what happened with the cubes when you added water. Now, as a hint, you want to talk about how the water maybe stuck to or affected your cubes. So you're going to have to take them out and investigate. Ooh, it's a little slimy. In my 2-centimeter cube, I have water on all sides of the cube. Made it a little slippery. Okay, it's actually dissolving a little bit. And now I'm going to take out my stack of smaller cubes. And I'm going to put them on the paper towel. And you can see they're coming apart very easily because every single side of the cube is wet. All of the cubes. So if I pull them apart, I'm observing what's happening with the water and the Play-Doh. And I'm getting water on every single side of my small cubes as well. So when you run the experiment, see if you have a similar thing happen and write down your observations. What did you notice when you put the water on the cubes and you took them out to take a view at all of it? Okay, Now, once you finish with that, we are finished with the Play-Doh. So go ahead and clean up your lab space. You can put your excess Play-Doh back, stick it in your lab box just in case we need it later. You can dump out your water and throw away your wet, mushy Play-Doh. Okay, now that you've cleaned up your Play-Doh, we're going to go through a little bit of math with our cubes. So underneath the observation box, I'm at number 6, and it says calculate the surface area and the volume for your cubes. Remember, area is a two-dimensional thing found by multiplying two measurements. In this case, we measure one side by the second side, or length times width. So we're measuring in centimeters, which means we will have centimeters squared for our unit. Volume, on the other hand, is a three-dimensional concept. We now added depth, so length times width times depth. And so we now have centimeters cubed, or to the third power, as our unit. Be sure to include the proper units for each calculation that you do. You want to first start with the surface area of 1 centimeter cube. So one of those small little Play-Doh cubes, what was the surface area, or again, the area of just the outer parts of the cube? You find the area of one side, and then you multiply it by 6 because there are 6 sides to a cube, right? Now again, surface area is the length times the width, which in this case is the same number, 1 centimeter times 1 centimeter, okay? And then remember to multiply that answer by 6 to get the surface area of the entire cube. The next one wants you to find the surface area of 8 1 centimeter cubes. So to do this, all you're going to do is take your answer that you just got and multiply it by 8. Nice and easy. Now you want to find the volume of your 1 centimeter cube. Now volume is going to be length times width times depth, but all 3 sides are the same for a cube, so it's really just your sides. So side times side times side, okay? Remember, this is a 1 centimeter cube. That is the length of one of your sides. And so this is going to be centimeters cubed. Now for your next one, the volume of 8 1 centimeter cubes. Again, you take your previous answer, you multiply it by 8. Our last 2 questions say the surface area of the 2 centimeter cube now. So now your side length is 2 centimeters. Find your surface area. Multiply it by 6 to encompass all 6 sides of the cube. And now find the volume of that 2 centimeter cube. So it's your length times your width times your height. Let me know what the volume is. Number 7, what do you notice about the surface area of 8 1 centimeter cubes compared to 1 2 centimeter cube? So they ended up being about the same size roughly. Okay, so what do you notice about the surface area? Look back at your calculations right up above and compare those 2 numbers. Number 8, what do you notice about the volume? So again, look at your numbers up above. What do you notice about the volume between the 1 centimeter set of 8 cubes versus the 2 centimeter cube? Okay. Now part D is going to go into some graphing. So at this point, you might be done with your egg lab, so you can jump to that. Otherwise, let's talk through some of the graphing together while your egg soaks in its colored solution. Okay, so part D, graphing surface area and volume. The ratio of the surface area to the volume is of the utmost importance. So when we say a ratio, you can think of it as like a fraction almost. So the surface area to volume, okay? That ratio, that balance, is going to be incredibly important. If the volume is too large, if our cell is too big, how can the cell membrane get enough good stuff in and enough stuff out? Okay. So when our surface area to volume ratio gets out of balance, the cell could actually suffocate, it could starve, it could drown in its own waste. Let's think about that for a second. So if a cell is very big, that means it has a very big volume, but a very low surface area. So the surface area to volume ratio are opposites, they flip-flop. So as the surface area gets big, the volume might go down. As the volume gets big, the surface area goes down, okay? So again, if the volume is too big, we don't have enough surface area or cellular membrane, and the cellular membrane is what lets things in and out of the cell, okay? So if the volume of the cell is too big, you might not be getting enough nutrients in, and you might not be able to get your waste out. We're going to look at this with some functions, with some math. So number 1, it says the surface area of a cube is found by adding the area of each side together, or multiplying by 6, which is what you did up above. So the equation for what you just solved is y equals 6x squared, where x is the length of the side of the cube, okay? Y equals 6x squared. Don't let that scare you. That's what you did up above. Now, you can take the cube and think about unfolding it into a net, which is the pictures to the right, and it's just a visual way of looking at a cube. So here you can see the surface area of the 1 centimeter cube when it's all spread out as a net. Again, the surface area is equal to 6 centimeters squared, or y equals 6x squared, where x is 1. Okay. Now next to it, we see the net for the 2 centimeter cube. So you can see the 2 centimeters, you have 2 blocks side by side for every single side of that cube, and the surface area equals 24 centimeters squared, or 6x squared, where x is 2. They want you to draw the net for a cube with a side length of 3 centimeters. So copy those 2 pictures up above, but just add a third 1 centimeter square to your net. It's going to end up looking like a cross. It's going to be wider than your other pictures, and you can use the grid provided on this paper. You then want to calculate the surface area. So if your equation is surface area equals 6x squared, where the side of your cube is x, you just plug in 3. 3 centimeters is the side of this new cube that you're drawing for number 2. Now it says the volume of a cube is found by the length times the width times the height, 3-dimensional. Since they're all the same for a cube, the volume is simply the length of one side to the 3rd power, okay? X times x times x, or x to the 3rd. Number 3, complete the table for the surface area and volume. You may use a calculator. So the first couple examples are given to you, and just go straight down, plugging in the side length for x. So in the column number 2, you're simply going to write out what the equation would be. So for number, let's say 3 centimeters, where we're starting there, for 3 centimeters in your second column, you're going to put 6 times 3 centimeters to the 2nd power, or squared, equals, write it out, 6 times 3 times 3. Okay, just like they did in the problem right up above. They're giving you an example that you can use as your template. Now in your 3rd column, give me the answer to that equation. Plug it into your calculator if you need to and solve it. Then you want to do the same thing for volume. So for your equation, volume is x to the 3rd power, or x cubed. So write that out. 3 centimeters cubed equals 3 times 3 times 3. And in your last column, give me the answer to that equation. Now the second part of number 3 says to highlight or bold the point of intersection where these numbers are the same. Okay, so look at your surface area answer column and your volume answer column. Do you see the same number for any of the side lengths? You should. Find whichever side length it is and highlight it or bold it on your paper. Number 4, using that table that you just made, you're going to make a graph. The x-axis, the horizontal axis, is going to be the side length of your cube, starting at 0, going up to 7 centimeters. Your y-axis is going to be the volume and the surface area. So you're going to be graphing both centimeters squared and centimeter cubed for your y-axis, and that's okay. You want to spread out the values, so make sure you have a lot of space for your 0 to 7 of your sides so you can see the graph nice and visually. Okay? You can either draw it on the graph paper, draw it on separate paper, and put it into your lab report, however you'd like to do it. Now again, you're using the first column of your table above for your x-axis, and then you're graphing both the third and the fifth column, surface area and volume, for your y-axis. Now your range for your y-axis is going to be pretty big. You want to make sure you encompass the tallest part, or the biggest number, of your surface area and volume columns. So I'm not going to tell you what that would be, but take a look at your table and make sure that you give enough room on your graph paper for your y-axis. You might not be able to count every single box, and that's okay. Maybe you go by boxes of 5, maybe you make every box 10, however you need to do it. But show that graph. Okay, it might be helpful to put the surface area in one color and the volume in another color, because you're going to be plotting both the surface area and the volume on the y-axis, right? Number 5, take a look at your graph. Think about what it's telling you. So it says, describe what this graph means in terms of the relationship between surface area and volume. So as your three-dimensional object or your cell becomes larger, what happens to that ratio between surface area and volume? If one goes up, how does the other respond, and vice versa? Right? We're on to your analysis. So at this point, let's go ahead and finish up our lab. So your eggs have been soaking in your colored solution for a little while. Again, make sure you go for at least 30 minutes, so pause the video and wait longer if you need to. And otherwise, let's go ahead and finish that up before you complete the analysis for the lab. Okay, so I just went and checked on my egg and my cube, and they're looking pretty interesting. I'm not going to show you how they look because I want to make sure that you do the experiment and can test it out for yourself without any bias to what happened with mine. But what you're going to do is you're either going to get a spoon and scoop out your egg and the cube, or you're going to dump it through a colander. Make sure you catch your cube though, but your food coloring liquid can just go down the drain. And next you're going to get a knife and you're going to cut both the cube and the whole egg in half. Take your ruler and have it on hand. So after you cut the eggs in half, you want to write some observations. Okay, so what happened? What do you notice? Is there any difference between the cube and the whole egg? If so, what is it? Number 5, it says for both the whole egg and for the egg cube, take your ruler and measure the distance in centimeters from the leading edge of the food coloring to the center of the egg. Okay. So measure the distance from the food coloring to the center. Put that in the table down below. Number 6, in which one did the food coloring reach closer to the center? Okay, so really which one has the smaller distance in your table up above? Number 7, remembering that the cell membrane allows substances the cell needs to enter and substances it doesn't want anymore to exit, explain why a large cell might have difficulty keeping up with their intake and output needs. So your eggs replicate a big cell and a small cell, okay? What did you notice about your food coloring water? Was it able to permeate inside both of them? Did it enter equally? Talk about the distance of your food coloring and how this might relate to a cell membrane, letting things in and out for a big cell and for a little cell. Okay. Now, once you get your answers down, we've already completed part C with our Play-Doh. We've already talked through our part D graphing. So we can now talk about the analysis. You've done all of your experimentation. Go ahead and pause the video while you finish all that up, and then we'll go to the analysis. So a lot of the analysis I'm actually going to let you do on your own so that you can come up with your own claims with the evidence that you made, right? But that's really what this analysis is for this lab. So using the information from part B, so from your egg lab, think of the evidence and make a claim about it, okay? What was your evidence? What did you notice? What did you notice about the food coloring between the big egg and the little egg cube, right? Could be just a simple statement. And what does it mean? So thinking critically, we're trying to think about our data, put it in terms of the body, and think about what it means, right? You noticed something. What type of claim can you say about cell membranes with big cells versus little cells? Question 2, you want to think about your Play-Doh cubes. So what did you notice about the Play-Doh cubes? When you poured water over them, what happened between the cubes? And how would that replicate one big cell in the body versus a lot of little cells in the body? Okay? Make a claim. Think about it in terms of cells in the body. What does it mean? Now, part D, you're going to use to answer number 3. So thinking about this net, the graphs, the numbers, what did you notice? Okay. What was your evidence between the surface area to volume ratio? If one went up, did the other go down? If one went up, did they both go up? What happened with the ratio? What did you notice? And what's your claim? So what does that mean? If a cell gets big, what happens between the surface area and the volume? If a cell is small, what happens with the surface area and volume? And how does that make it any easier or harder to get nutrients in and out of the cell? Now, number 4, we're tying it all in, okay? So everything that you've thought through and you've worked on between your eggs and your Play-Doh and your graphing lab, it says go back to your share your thoughts at the very beginning, okay, before you did your research, before we did your experimentation, and think about what you've now learned. Did any of your answers change? So it says rewrite your response in the space provided. Include at least one sentence of supporting evidence from your lab. Okay, from whichever part, include at least one supporting sentence of evidence that you found today. And then write one sentence that summarizes the overall statement. Okay, so you want to write down your hypothesis, what you thought at the beginning of the lab. You want to then write down the evidence you collected, and then you want to write down a summary of what it means. So this exercise here is not only pulling all of the information together that we've done today in the lab, but it's also starting to have you think in terms of writing a scientific paper, okay? Writing an analysis. So you thought one thing might happen, you've now collected data, which you need to report all of, and then you need to come back and think, well, how did that data shape my hypothesis, okay? The scientific method. You might have a few paragraphs here. You don't need to write a whole page. You don't need to include every single thing in the lab that you did, okay? But give me some good examples of what you thought, how your evidence matched up, and if your hypothesis was correct or not, and why, okay? So again, maybe just a couple paragraphs at most. Try to be succinct or concise. And after you finish that, you're finished with the lab. So I hope you have fun, and as always, let me know if you have any questions.